Hi guys, so today we're going to be solving practicals paper, variant 4, May, June 23. Okay, so it says milk is a source of protein and reducing sugars. Different types of milk contain different concentrations of protein, and the concentration of protein in milk can be measured using potassium hydroxide solution and copper sulfate solution. So they gave us a milk containing 5% concentration of protein, distilled water, potassium hydroxide solution, copper sulfate solution, and milk containing an unknown concentration of protein. That's you. Okay, so first of all, it asked us to carry out serial dilution of milk containing 5% protein to reduce the concentration by half between each successive dilution, and we need to prepare four concentrations, and after each we have to have 10 centimeter cube left in every beaker. Okay, so first of all, we know that serial dilution by half, it means that we need 1 to 1. It means the ratio is 1 to 1. Ratio of concentration to water. So 1 to 1, the total is gonna give us 2, which is half, which is half. Okay, so we have 20 centimeter cube of 5%, okay? And then he added 0 water, and then 10 was left. That means he removed 10 centimeter cube of 5.0, and he added 10 water. So by that we got 10 centimeter cube of 2.5, and then we removed 10 centimeter cube of 2.5 and added 10 water, and then removed 10 centimeter cube of 1.25, which is the new concentration that we got. And then we removed 10 centimeter cube of 1.25 and added 10 centimeter cube of water. So that by that we got a 10 centimeter cube of 0.625%. And we did the same to the last beaker, but in the last beaker we have 10 and 10, which will give us 20 centimeter cube of 0.3125. You can either leave it as 20 or make an arrow and see discard 10 centimeter cube. Okay, so now it's telling us to prepare the concentrations, label the test tubes, okay, and put 2 centimeter cube of each concentration, and then put 2 centimeter cube of K, which is potassium hydroxide, and 2 centimeter cube of C, which is copper sulfate solution. Okay, and then we need to observe the color in each test tube. Compare the colors to this figure. Okay, so we know that percentage concentration of protein. So as the percentage concentration increases, like the protein increases, obviously the color is gonna be darker. Okay, so here I did a percentage concentration of all and a color intensity, and I did trial 1, trial 2, and mean. So 5% had the highest, darkest color, which is dark purple, dark purple, dark purple, and then 2.5 is gonna have the least, and then 1.25 is gonna have less, and 0.625 will have less, and 0.325 will have the least color, because it has the least concentration of protein. Okay, and then it asked me stir U and put 2 centimeter cube of U, and you guys should be sure to read the volume that he kept here. So he said 2 centimeter cube of each concentration, because then for the unknown he'll ask you what how much amount. In any other paper he can ask how much amount of U you have to put, and then you'd say 2 centimeter cube. Okay, so then we'd carry out the investigation on you, but you obviously don't have to. For the unknown, you can put it either as one of the three. You can put dark purple or purple, okay, or anything. So I put purple, and it can be 2.5 or 1.25, and I choose 2.5. okay, and then it says describe one significant error is step 7. Step 7 is observe the color. So you're gonna see color change is subjective, you can't judge the color change by eye, so you can use improvement is use colorimeter. okay? And then it says suggest how you would modify the experiment to determine the concentration of reducing sugars in the sample of milk. So for reducing sugars, you're gonna use Benedict's solution, and then you would heat it to 80 and measure the time for the first color change, or you can compare the results to a standard solution. So here for graphs, the one on the left is always x and the one on the right is y. Okay, so it asked us to draw a bar chart. So here's the answer. Okay. And then here, one of the reducing sugars found in cow milk is lactose. Some people are intolerant to lactose and cannot digest it. A method of producing lactose-free milk is to treat cow's milk with enzyme lactase. Lactase hydrolyzes lactose to produce glucose and galactose. A scientist investigated the effect of different lactase concentrations on the mass of lactose converted to glucose and galactose in 10 minutes. Okay, so it says use the graph in figure 1.4 to determine the mass of lactose converted when 0.16 mole is used. Okay, so then it says determine the mass of lactose you converted when 0.16 is used. Okay, so I came here, which is 0.16, and then you guys should indicate it on the graph, and then I got here 54, and then to find the rate you're gonna divide 54 by 10, which is 5.4, because it said like the total is 10. Okay, so it says explain the shape of the graph between 0.1 and 0.2. So this is 0.1 and 0.2. So between 0.1 and 0.2 there is an increase. Why? Because the concentration of the enzyme increases. So we're gonna see concentration of enzyme increases, and as the concentration of enzyme increases, we're gonna have more free active sites. So with more free active sites there will be more successful collisions. So with more successful collisions, we're gonna have more enzyme substrate complexes. Okay, so now it's asking us to draw a slide through N1, and this was the picture. So it's asking us to do this part, which is basically like this part, okay? And this is a stem, and I can see that it's a monocotyledous stem because the vascular bundles are scattered. Okay, so let's start by drawing. First of all, we have to draw the epidermis. Okay, this is, and then the two lines have to be close and like consistent thickness. Like, don't make the broken lines, okay? And then a root will have another section, which is the cortex. So I'm gonna draw another line, which is like kind of for. Do it again. Okay, and now I'm gonna draw the vascular bundles. I can see one here. Okay, and the big one is the xylem, and then this is the xylem, and this is the phloem, and between them is cambium cells. Okay, and then I'll draw another one. Xylem, phloem, cambium cells. Okay, and then another one. The xylem should like take most of the space. It should have like the bigger part of the drawing. Okay, and then another one. Like, this is not the best thing, but you guys can do better. And it's asking us to label the xylem. We can label anyone. For example, I'm gonna label this one. This is the xylem. Okay, so now it's asking us to observe cells in the central tissue of the stem and draw a group of 4. Each cell must at least 2 other cells. So central tissue is basically the parenchyma cells, okay? Like it's this space right here. It's basically like the cortex cells. Okay, so we're gonna draw a group of 4. Let me draw. And then I'm gonna draw one right here. They have to be like different sizes because like not all the cells are the same size. Let me draw. Okay, and now you guys have to draw the cell wall. It has to be close. Okay, and it asks us to identify the cell wall. So this is the cell wall, and then 2 cells should have a nucleus, and like the others not, because the nucleus won't be visible in as okay. So now it gave us figure 2.2 is a photomicrograph of a stained transverse section of a stem from a different plant species to the stem on N1. So this one is different from the other one. Okay, and now we have to identify 3 observable differences. Okay, so first of all, I can see that this one has hairs, but then this one doesn't. Okay, so we can see hairs, or you guys can see trichomes. Okay, I can see in N1 it's absent, and then on figure 2.2 it's present. Okay, and then we can see the size of vascular bundle. This vascular bundle are bigger than these. Okay, so then we can see size vascular bundle. This is smaller and this is larger. Okay, and then we can see the number of the vascular bundle. I can see that N1 has more vascular bundles than figure 2.2, which is like 1, 2, 3, 4, 5, 6, 7, 8. That one is obviously more. So here is more and here is less. Okay, and then lastly we can see a shape of stem. I can see that this one is triangular shape and the other one is rounded. And lastly, don't forget to, the other mark is on like making the lines on the table. Okay, so now it gave us part C. It's telling 2C, figure 2.3 is a stage micrometer calibrated using an eyepiece graticule, and then it's telling us that one division on either the stage micrometer scale or the eyepiece graticule is the distance between 2 adjacent lines. So the length of what division on the stage micrometer shown here, it's 0.50. So the distance between 2 adjacent lines is 0.50. So now it's asking us to calculate the actual length of one eyepiece graticule unit shown in figure 2.3. Okay, so I can see that from here from 90 to 50 is gonna be 40 units as like 10. We have here 10, 20, 30, 40. Okay, so from 90 to 50 is 40, and then 40 is 0.50 millimeters. So we need to find one eyepiece graticule. We're gonna calculate this. we're gonna get 0.0125 millimeter, and then we need to convert it to micrometer. So we need to multiply this answer by 10 to the power of 3, and then we're gonna get 12.5. Okay, so now it's telling us the figure 2.4 is a magnified section of the stem showing figure 2.2. This was taken with the same microscope and the same lenses used to make the photomicrograph in figure 2.3, and it's asking us use the calibration on the eyepiece graticule unit from CI to calculate the actual length of the trichome labeled X. Okay, so we have this trichome. It starts from approximately here, which is 44, and then I can see that it ends like here, which is 79, and then we're gonna do 79 minus 44, which is 35. You guys can get a different answer because there's a range. Okay, so we have 35 eyepiece graticule units for X, and then we have one eyepiece graticule unit, we got it for 12.5. Okay, so we're gonna do 12.5 times 35 eyepiece graticule units divided by 1. Okay, the answer is gonna be 437.5 micrometers. Okay, and like this we're done with the paper.